Okay, well, thanks very much for having me here. Today, I want to talk to you about how we're using big data to really improve personal health. So I'll start with a premise that many of you may agree with, which is that medicine as practiced these days is highly flawed. It's flawed in actually many, many different ways, but these are a few listed here. First, we're very focused on treating people when they're ill. As such, it's very reactive. When people are healthy, we measure very few things, and also they're measured very infrequently. But perhaps the biggest problem is that nearly every decision about your health is based on population-based measurements. Instead, most of us would agree that we should be focused on keeping people healthy, as such be very proactive. We're certainly capable of measuring many, many more things. The frequency, I would argue, should depend on what you're at risk for, which can come from a personal genome sequence, but certainly can come from trajectories of other markers as well. But perhaps most importantly, we should be focused on really individual-based medicine rather than population-based medicine, and as such, be focused on precision health. And just to illustrate this point, you've probably been told since you were small that your temperature is 98.6 when you put a thermometer in your mouth, a healthy temperature. But actually, if you look at the data that's out there, this is one of a number of studies. The numbers, first of all, it's off. It's more like 97.5. I personally am 97.3 most of the time, and it's dropped a little bit over the last 8 years. But more importantly, there's a range. So this is the 25th quartile at 94.6. This is the 75th quartile at 99.1. So in today's world, if your normal baseline temperature is 94.6 and you go to a physician's office, what'll happen is your temperature will, you go to a physician's office and your temperature is 98.6, they'll basically say, well, you're normal, you're healthy, but you're really at 4 degrees Fahrenheit. You're really probably not healthy if you're 4 degrees higher than normal. And so that's why we would argue you should really know your baseline parameters so that you can find illness at its earliest possible time. And that's a theme I'll hit on several times throughout this talk. So your health is comprised of many factors. It's certainly due to your DNA, but many other things influence your health as well. Pathogens, exposures, talk about that a little bit today. Stress, food, exercise, these all contribute to your health. We know that intuitively, but we're in a world where it's now possible to quantify this, sometimes well, like activity, sometimes it's tough, like food, but still doable. But you can certainly quantify the impact of these various things on your health by taking deep molecular measurements, we believe. And we think this will be very powerful for following people's health into the future. So as many of you may know, a number of years ago, we set up something we call personal omics profiling, where the idea is to take deep data measurements on people. So we sequence their genome once, and we follow them many other omes as well in its most deluxe form, 14 different omes. Epigenome, meaning DNA methylome, transcriptome, these all come from your peripheral blood monocyte cells, your immune cells. But many of these other measurements, your proteome, your cytokines, metabolites, lipids will come from plasma. Microbiome, we measure gut and nasal, and more recently we have tongue and skin data. And we do this on top of deep clinical testing, questionnaires, and some advanced tests as well, like stress echocardiograms. And about 8 years ago, we'll spend some time on this. We do a lot with wearables and biosensors. So one aspect of this is taking very, very deep data on people where we make millions of measurements, but the other is we do it over time. So we sample people roughly every 3 months when they're healthy. And then when an adverse event comes along, like a viral infection, very relevant to today's world, we'll take many more samples, excuse me, typically 5 to 7 additional samplings. And again, we're trying to do this over time. We've been running the study, well, on me for 11 years, but for most people, it's about a little over 8 years, some a little more, some a little less. But so you may say, why are we doing this? Well, we're trying to understand what it means to be healthy. How does that change over time? How does that differ between different people? What do people's different molecular measurements look like? We also want to understand what happens when people get ill at the earliest times possible. And lastly, we're very interested in seeing whether these advanced technologies can be better used to manage people's health. And so with regards to the last point, we actually, from the first 3+ years of study, we found 49 major health discoveries, and it spans a wide range of different areas: heme oncology, cardiovascular disease, metabolic disease, so on and so forth. And it was no one technology that actually found these things. So first of all, about half the people had something. And we think the reason that number is high is because it's an older cohort, 53.4, although it is a pretty wide range. And as I say, it was no one technology that would uncover these things. Sometimes it was genome sequence, sometimes imaging. Usually it was combinations of things like imaging plus biomarkers that went up in the blood and so on and so forth. But to give you a few examples, we did catch some with early lymphoma, 2 people with pre-cancers, one called MGUS, another smoldering myeloma, 2 people with serious heart issues, one from Genomics, talk about in a minute, another from a wearable device, quite a bit on diabetes and so on and so forth. So again, a wide range of different things were found and different technologies unveiled different conditions. So the genome was quite powerful. 13 people learned something important about their genome, 12 from their Mendelian risk, if you will, and 1 actually had a pretty important pharmacogenomics, and some people even learned important things through their polygenic risk scores. So the 12 that learned something from a Mendelian condition, these are highly penetrant genes that with some frequency could put you at strong risk for certain conditions. And the most famous one, I'm sure you know, are the BRCA mutations. 1 individual in our cohort, this is just the first 70 people, did in fact have a BRCA mutation. And other people had different things. These are what are called the ACMG genes. And these are other genes. We actually look at the genome quite thoroughly. And it turns out 3 of these people learned something pretty important. So this individual, turns out pretty young guy, has a mutation, the RBM20 gene, which puts him at high risk for cardiomyopathy. And it turns out his father died of a heart attack in his 60s, aunt had one in 50s. And then as a follow-up to this, he did a stress echo, and sure enough, he actually has a heart condition, is on medication. So, and this was caught at a pretty young age. So this is, we would argue, quite useful. This individual also fairly young. There were 2 of them. One of them, they both had mutations in the SDHB gene. This puts you at high risk for certain kinds of cancers, and 1 person did a whole body MRI follow-up. Sure enough, actually had early thyroid cancer, and the malignant part was removed. Was able to keep the rest of the thyroid, and you know, it's doing fine, doesn't need thyroid replacement therapy or anything like that. 9 individuals coming into the study were thought to be type 2 diabetic. 1 of those actually turns out to be a MODY mutation, which is a different kind of diabetes, and it's been on the suboptimal medication for years. And the others are getting monitored for their various conditions as time goes on. So the point out of all this is that genome sequencing did uncover a number of important things. I won't be able to show you all the other things like lymphoma and things we learned, but I will tell you that we're spending a lot of time on type 2 diabetes and insulin resistance, which is associated with that. So coming into the study, 9 individuals thought they were type 2 diabetic, but it turns out there were 2 individuals who were type 2 diabetic who didn't know it. So we did uncover that. And a lot of people were actually pre-diabetic, meaning they were elevated to hemoglobin A1C, which is a marker for diabetes. And they didn't know it, and that's very common. 9 out of 10 pre-diabetics don't know it, but 70% of them will go on to become diabetic in their lifetime. So we think this is a big deal to actually know this. Now, we're very interested in how people become type 2 diabetes. I forgot to say I'm type 2 diabetic. I actually figured predictive from my genome sequence, and God, as you'll see in a minute, as we were profiling me. But during the course of the study, we discovered 9 individuals became clinically diagnosed as diabetic, and others had diabetic range values. And so we're very curious, how do people become type 2 diabetic? Do they just gradually get there? Do they, you know, does something trigger it? What's going on? Well, of the 9 that became clinically diagnosed as type 2 diabetics, 2 gained weight, sort of a classical way to get diabetic, and the microbiome diversity dropped, that some of them went on metformin, and so on. But the other 7 individuals didn't gain weight at all. 5 of the 7 actually slowly became diabetic. So they just gradually, by either fasting glucose, which is shown here in orange, or hemoglobin A1C, this individual just gradually became diabetic, then ultimately went on metformin and got under control. This individual got there through an oral glucose tolerance test, which is sort of what happens when you drink glucose, and basically, again, gradually got there through these different measurements that was picked up. And these measurements pick up different aspects of glucose dysregulation. So again, these folks, they didn't gain weight, but they gradually got there through age. 2 other people basically got spiked into diabetes, if you will, as though something triggered it. And I'm one of those two. This is actually me in about 8 years of data. I first became type 2 diabetic after a very nasty viral infection, respiratory syncytial virus. I know this looks like a blip, but it's actually about a year's worth of data. And I had to do a lifestyle change to get it under control. I did get it under control initially. In fact, it was so boringly low, I stopped looking at it. And then someone else spotted it here. It's a Mike, your hemoglobin A1C is 7.0, which is, again, that's diabetic range. So he sampled me a few times. It turns out I had stopped running, and guess when that was? That was here. I also had a viral infection, so it's not clear what triggered it. But I did start running again. I brought it down. I never got it down to totally normal levels. It kept creeping up, and ultimately I did go on metformin. Turns out I'm a non-responder, which is very disappointing. In fact, my it's not shown here, but my hemoglobin A1C got all the way up to 7.5. And finally, the solution for me was more data. It turns out I'm insulin sensitive. We can show that by a variety of means, and also make insulin just fine. So what's wrong with me? Well, it turns out the answer is I don't secrete insulin from the pancreas, and you can show that by a variety of ways. One is a measurement called glucose disposition. So in fact, if I do have glucose, it will pile up, if you will, because my insulin doesn't get released from the pancreas. And it turns out there's a drug you can measure that. There's a drug for this. It's called a Prandtl derivative called rapinolide. And when I take that, it turns out I'm a good responder. So the bottom line out of this is we call this precision diabetes. By better knowing exactly what was wrong with me, I could take the right drug. I don't respond to metformin. I do respond to this. And it turns out I also respond to something called SGLT2 inhibitors. So that seems to be working as well. And these tests, I know they sound sophisticated, and some of them are, but they can actually be done pretty easily. We can be doing this for essentially everybody. And so by better understanding how people are again, have problems, I think we can better address them appropriately with better data. Now, one of the things we learned by doing this deep profiling is that people's personal profiles are actually fairly stable. It doesn't matter which one we use, your personal measurements actually, they don't drift all that much. So this is just showing 12 people with 10 healthy visits. Each dot's a different visit, and each color's a different person. And I'm the light blue one. And there's like 6 years of data here for me. And you can see no matter which test you use, my dots all cluster together. And it's true even for the transcriptome, if I were to show you this in 3D. Again, people's profiles, whether it's metabolome, proteome, what have you, clinical labs, they're fairly stable, they're fairly personal. And they don't drift all that much. Not only that, if you get a viral infection or you do some perturbation, in this case, this is an exercise study where we had people run to their VO2 max, you will shift your profile, whether it's a viral infection, whether it's an exercise bout, whether it's the weight gain, weight loss study, you shift your profile, but you'll still look more like you than anyone else. Again, these measurements are very personal and they're very deep. And we think this is a big deal. And this is why this topic you have, personal genomics, is so important, because I think there's a mindset out there that you need thousands of cases in order to see a difference between disease and normal. And that's true if you're comparing across populations, across individuals, because the difference between people is often greater than the difference between disease and normal. So it's actually hard to tell difference, again, when you're working at population levels, but you can see it easily at a personal level. If you have a healthy baseline and you shift, you can't miss that, whether when you get a viral infection, when you do an exercise bout, whether you gain weight or lose weight, you can easily see that shift. But it's hard to do it when you're comparing you against 500 other healthy people, because the difference between people is too large. That's a hard concept for a lot of folks to believe, to be honest. But it's really what personalized medicine, personalized health is all about. That's shown here. Each cluster is a set of people who did an exercise bout, and all their time points cluster together. They ran to the VO2 max. All right, so I told you patterns are pretty stable, but it turns out there are two kinds of differences we did find. One is seasonal drifts, and the other is actually with time, aging. And so one of the things we asked is, we have all this data out there, and we were thinking about it and saying, well, gosh, I wonder how many biological seasons there are, because you've been taught since day zero, there are 4 seasons, they're 3 months each. And if you think about it, of course, that's hogwash. Maybe there's 15 seasons, maybe there's 3, who knows? Why would they be 3 months each? That makes no sense at all. Maybe some are 2 weeks, some are 20 weeks, who knows? So we let the data tell us what's going on. How many patterns are there in the data? And so the answer is we had sampled people throughout the year, taking many, many measurements over a number of years. In this case, about 4 years of data. And it turns out there's 1,152 molecules of these thousands, about 20,000 molecules of and on top of that, microbes we're following as well. And we asked which ones are showing seasonal patterns. And the number is, it's about 1,150. And some of these are well known. Hemoglobin A1C is a well-known case. Even red blood cells are known to drift. A lot of the stuff was brand new. Here's a circadian thing, which is kind of cool. And some of the microbes actually show seasonal patterns as well, which is pretty neat. Okay, so we had these 1,150 some odd things drifting, showing seasonal patterns. Then the next question is, well, how many major patterns do they fall into? Is it 15? Is it 3? Is it what? And the answer is it's 2. And I forgot to say these individuals are all from Northern California. A few are from Southern California, 3 of them are, but most are from Northern California. It's 109 people. I probably forgot to say that. Anyway, it turns out there are 2 patterns, and we can show this mathematically for those who follow these elbow plots. But and one of them is in winter, as you would expect. So you probably say California. Of course, there's 2 seasons, nice and warm out there. And one of them is in winter, and that's probably what you would guess. So a lot of molecules kind of come up late in the year, persist into January, and then come back down in summer, so on. And then the other one, you might say it should be summer, but the answer is no. It turns out it's April, May. It's sort of late spring, if you will. Well, depends how you define spring, but it's basically late April, early May when a lot of molecules go up. And these patterns, it turns out, make sense. So this one here turns out is when viral pathways are getting induced. So this is when people often get sick over winter, as I'm sure many of you discovered through the pandemic. And you see acute phase signaling, even acne is known to peak in winter. This one here also makes sense to a large extent. It's allergies and asthma popping up again in April and May. But there's a lot of other markers here too: cardiovascular disease, metabolic disease, type 2 diabetes. These all peaked around this time too. And in hindsight, that does make sense. It turns out even in California, it does rain here. It gets colder, 60 degrees cold. That's cold for us. And the bottom line is we think people are less active during that time. And so a lot of these molecules gradually accumulate and then become, at higher concentration come April, May, and then spring comes and people are more active, it goes down. So this also makes sense in hindsight too. Some of it was known, a lot of it wasn't known, and some of this other stuff I don't fully understand yet. So anyway, the bottom line is there are 2 patterns in California. They also have clinical markers. Even your microbiome will shift with these seasonal patterns. So you'll see a number of clinical markers peak late April, May, and then others will peak in winter. And again, that makes a lot of sense for what some of these markers are. And we think this is useful and potentially actionable. If you see things drifting too low, you might actually want to warn people, you're getting outside your normal range, winter's coming, maybe you should, eat better or exercise more, do more stuff because you're heading in a bad direction. Likewise, maybe when late spring comes, you can say, well, spring's here, you better get moving because your molecules are not so good. And as I say, your microbiome changes too. That's a fascinating story as well. The other time when people's markers change is over time, i.e. with age, as we're defining it. And because we've been sampling the same people over time, we can actually see how they're changing at a personal level, which hasn't really been done before. We think you need 5 measurements within 2 years or less, and you can actually see what people's aging pattern is. And this one over here is me. There's about 4 years of data. And I'm a pretty typical ager. My coagulation pathways, my metabolic pathways all go up with time. This individual, though, their top pathway is cardiac hypertrophy signaling pathway. So they're actually a cardioager, if you will. Their top pathway that's going off is heart. So the way we think about this is like, cars that the whole car is getting older, but some parts are wearing out faster. And you can see this. So we asked, well, just like seasons, how many major patterns are there? We had 43 people. It's not a lot, 43 people that we had enough data on. And it turns out they fall into 4 major classes. We call them ageotypes. Now, I know there's going to be more than 4. It's just that that's how many we were statistically empowered to find. And so each column here is a different person, and each row is a different ageotype. So kidney ager, liver ager, metabolic, immunoager. And so the folks over here, they're aging in all 4 categories. This person's aging in 3 of the 4. I'm somewhere in here. I'm not much of an immunoager, but I am aging, I think might be this one, in some of the other categories. This person's a kidney ager, kidney and metabolic, and so on. So again, different people are aging differently, and we would argue these are the parts that are most acute, having problems. Not only that, you can actually look at clinical markers associated with these ageotypes. They're just showing 6 clinical markers that people follow, and it's very informative. So for example, the group as a whole goes up for, say, hemoglobin A1C. That's known. Older people have more hemoglobin A1C than younger people. That's how people used to study this. So the group as a whole goes up. Well, what we found is that, of course, there are some people go up. A dark line means it's statistically significant. And for 4 people, they actually were going up with time. But what's kind of cool is that 4 others were, in fact, going down. And in a significant fashion. This is an easy one. We could see what was going on there. And in 2 cases, they went on a diet. 1 person did exercise. 2 people lost weight, and so on. So we could actually track people's patterns. Now, in this case, and so again, we could associate it with lifestyle. Here's a case with creatinine, which is an indicator of kidney function. The group as a whole was going up, which we would expect, but 10 individuals went down, and 8 of the 10 are on statins. And we don't fully understand this. They didn't, as far as we can tell, change muscle mass. So maybe there's something beneficial about statins when it comes to creatinine function. That's something we're looking into. So the point out of all this is that we can follow people's personal aging patterns, and then we can also follow the rates. I won't show you, but some people, their biological aging is faster than their chronological aging, and they're aging, as I say, in different ways. Other people are actually doing pretty good. Their slope is kind of slow. Their biological age is actually less than their chronological age, which is kind of cool. So we think, again, we can tease out people's ageotypes, and we think this information is actionable. Now, the other thing we've been doing a lot, one thing people ask, well, Mike, so this is all nice and amusing. So what? How are you going to scale this? What you're doing is very, very expensive. So I'm from Northern California. So what you do is you start a company. We did totally conflicted. It's called qBio. And what it's doing is it's actually doing a medical version of what I showed you. It's doing deep data collection on folks, medically relevant data. And it also has a whole body MRI, which turns out to be pretty powerful as well. And just from the first 100+ people, we've discovered a lot of important stuff, caught some with early prostate cancer, ovarian, even pancreatic cancer, caught some early with that, cardiovascular, and so on and so forth. And sometimes it was caught because we had multiple lines of evidence. And sometimes it was caught because we had these longitudinal profiles. You could see the change that was going on in people's profiles, and that said something was up. And so we think this kind of approach will be very, very powerful, and I hope will be very useful for transforming the way we do medicine now, measuring people when they're healthy, and then later, if you catch something, you can act on it before they have symptoms. Now, along these same lines, we got very, very interested in wearables. We got interested at a time when people were mostly using these as fitness trackers. Apple Watch didn't exist. And people were, they put these on, they wear them for 3 months and throw in a drawer because they kind of understand their patterns. They were done with it. But we realized these are pretty powerful physiological monitors. And they make hundreds of thousands of measurements every day. Some of them now make as much as over 2 million measurements on you every day. And so they're powerful because they're tracking you 24/7, 365 days a year. They're collecting a lot of physiological data on you. And in the US, they're worn by millions of people. 20%, 50 million people wear a smartwatch, which is a lot. And it depends on the watch, but they can measure all kinds of different things: heart rate variability, respiration, changes in blood oxygen, skin temperature, so on. So even blood pressure, it's kind of shaky in the measurements, but it does measure some sort of semi-relevant parameter. So the way you use these is you wear them, you figure out people's circadian patterns, and then you look for deviations from that. And so what we discovered actually while we were doing this is well, first of all, people's blood oxygen and drops on airline flights and their heart rate goes up. Most people don't know that. Actually, as far as I can tell, only pilots seem to know it. But we also discovered that these are very, very powerful for measuring illness and infectious disease. And in fact, I discovered my Lyme disease from this. And the story there was I was helping my brother put up fences in rural Massachusetts. 2 weeks later, flying to Norway through Germany. And on that last flight, my blood oxygen was abnormally low and my heart rate was high as measured by a smartwatch. And then we went back and looked at all the data I had. It was 2 years of data. And there were 4 times I was ill, one of which was asymptomatic. We only knew I was ill because I had high C-reactive protein, a sign that you've been infected. So I was asymptomatic. And every single time I was ill, I had a high resting heart rate. This is shown by outline measurements. You can show it by median values. This is just a better way to show it. And also it turns out, this is all retrospective, I discovered my skin temperature was up, and I figured out Lyme time too, but retrospectively. So we wrote an algorithm to be able, so point out of this is that every single time, including the Lyme time, I was ill, I had high heart rate and high skin temperature. In the case of Lyme, it really did help me. It's how I first got clued in that I actually had the illnesses before symptoms, and later got symptoms, went to a doctor. He recommended penicillin, and I told him I need doxycycline. A little tense there, but anyway, we got it all cleared up. He gave me the doxycycline. So, and it was truly Lyme positive. I mean, I was tested when I got back in, and yeah, I gave blood. It was negative before I left. So bottom line is, we then wrote an algorithm we call change of heart that follows your baseline and looks as a delta plot, looks for a change in resting heart rate. And it turns out it works. Every single time I got ill, we could pick up this change in heart rate, including the asymptomatic time. Every single time we picked it up prior to that, that was one of these 2 times, prior to symptoms. And it worked on me, it worked on 3 other people, one of whom got sick twice. So then the pandemic came, as I'm sure you all know, and we'd been improving our algorithms, building up an infrastructure to do this at scale. Pandemic came, and of course, it's really been hitting hard. And even now, where we're still running around 60,000 cases in the US, this is US cases. And I know it's higher in many parts of Europe. So we're certainly not out of the woods yet, still running very, very high. And PCR is just a terrible way to do it. You get results next day, whereas smartwatches can follow you in real time. So we set out to test whether we could detect this. We had an infectious disease study running. We modified it for COVID-19, at least the IRB made us do that. So anyway, then we launched the study, partnered with Fitbit, Garmin, and others, and right away enrolled 5,300 people, 32 of whom are what we call golden data sets. They're basically wearing, in this case, a Fitbit during the time they got ill. They have a diagnosis date and a symptom date. And so he first said, what can we see when people are wearing these watches? And the answer is you can actually detect it pretty easily. This is our very first case. This is just looking at resting heart rate. Here's a diagnosis date here in purple, symptom date in red. And look when they first got ill, basically. This is a resting heart rate. It jumped up 9.5 days prior to symptom onset. You can't miss that signal, very, very clear. And I'll show you in a minute, we have an alarming system for picking this up. So this person presumably was running around spreading the illness for 9.5 days, not knowing it because he was asymptomatic. So we went on and showed that basically 26 of 32, so 81% of folks, we could see this elevation of heart rate. It doesn't work on it for everyone. We have a hard time getting a stable baseline on certain folks. So it's not working on everyone. 3 of those 6 we missed had severe asthma. I think it's just hard to get a stable baseline on them. But anyway, on the whole, we could show that we could detect COVID, this jump up in resting heart rate, 4 days on average before symptoms, 7 days before a diagnosis. So we could get this early jump up in resting heart rate well in advance of symptom onset. And it's not just specific for COVID, it also, as you might imagine, when other illnesses will have the same effect, there the median's 2 days before illness. And that makes sense because, so these are non-COVID cases. That makes sense because COVID has a longer pre-symptomatic period than do other illnesses, so 4 days versus 2 days. The average heart rate, by the way, is about 7 beats per minute that people jump up. So then we wrote a real-time online detection system. This is all retrospective, where we can see when your heart rate first jumps up, and try with the goal of setting off an alarm indicated in blue prior to symptom onset. And so we built this thing, and it does work pretty well. I'll show you in a minute. It's not perfect by any means. We've tuned the alarm to go off every 2 months or so on average. And we picked that because we think that's a frequency that's still pretty sensitive for detecting early onset. And it's not going off too often that people can't deal with. There are other things that trigger the alarm, as you'll see in a minute. So the bottom line is, here's the first person with COVID, again, retrospectively, 9 days ahead. I think this is 7 or 9 days as well, another COVID time. There are other bumps, as I say, just the way we've set this. These are real jump ups in resting heart rate. And 4 of 7 people have what we call a holiday bump. Now we know alcohol, high altitude, travel can do this, not 2 drinks at dinner, but really tying one on will lead to an increase in heart rate. And so different things do trigger this. You have to contextualize the information. So I don't know if this is in-law stress or alcohol or travel that triggered this. Is before the pandemic from a year earlier. So, but there are these other event trigger happenings going on. So the bottom line here is that here's another illness. Here's a healthy person with a trigger. I don't know if that's asymptomatic stress, what have you. Now we're cataloging all that. So back in the beginning of December, we now set up a real-time alerting case. All of you guys can join, no matter where you are in the world. You do need to bring your own device, Apple Watch, Fitbit. And what we've done is set up this online alerting system where when we see your heart rate go up, you basically will get a signal like a red alert, and then we want you to log in what's going on. And it does work. This is a case where the alarm went off here. Then 5 days later, their symptoms appeared, and they got tested and were positive. And it works about 70% of the time. Again, it's not perfect. We'd like to pull in high resolution data, other kinds of data, which will be more powerful. This is a bit dated. We're over this now. We're about 3,500 participants. Here are the numbers. 44 received an alert of 63. And not only that, we actually found 3 people were asymptomatic who actually had a big jump up in their resting heart rate. So we know it works for asymptomatic cases as well. So we published the first version of this. This is a little dated, I guess, but back in November. And again, we published it on our Fitbit data. Eric Topo and Stephen Steinhubble published their Fitbit. They didn't have the alarming. They have just showing that you can detect this. And it does work for other devices as well. Oura Ring, a study came out on that, Apple Watch, and so on. I forgot to say our online detection works for Apple Watch, Garmin, and for Fitbit. So we have this working now for a lot of devices. We have a slightly different algorithm I told you. I don't think wearables are only going to be used for just infectious disease detection. I think they'll be powerful for that. But they actually give other clinical information as well. So we have a paper coming out that where we did machine learning and show that other kinds of measurements, as hematocrit, red blood cells, so on, can get picked up at some predictive level with wearable devices, with smartwatches. And it makes sense because these things are actually following certain things like they're doing spectroscopy on your hemoglobin, so they can actually pick up some of this. And some of the measurements they make actually don't have a clinical correlate like galvanic stress response, but we think it'll be very powerful for physiological measurements as well. So we've built a dashboard to be able to pull all this data in, the wearables, microbiome, clinical lab data. It could display it at whatever resolution you want, daily, weekly, monthly, yearly. And then we're now in the process of building these algorithms to follow your health in real time. This is one for me, picked up when I got infectious disease, picked up my bike accident. I don't think you need a smartwatch for that, but we think it's going to be powerful for things like chronic fatigue, things of that sort. So we're building models around those now as well. And so this is Mike Snyder's world. I envision a world where people who are getting their genome sequenced prior to birth, and along with biochemical and wearable measurements, we can better follow people's health with the ultimate goal of predicting disease risk, catching disease early, and monitoring treat disease using big data. So I have an amazing team. I'll just have to wrap up by saying, when you, Brian, Kevin, Tejas all spearhead a lot of the omics, George Weinstock on the microbiome, Trace McLaughlin, and then the folks building a lot of our health dashboard of wearables, Chow, Lee, Jesse started it, but now we have a pretty big wearables team rolling this out. Please join our study, innovations.stanford.edu. And I'm happy to take questions if you have any.